Okay, guys. So we are starting with the question on slide. As you can see on your slide file, the question says, it is from the year May-June 2005, paper 3, right? The question says that you've been given a slide, K1, which is a stain transfer section through part of a dicotyledonous plant. I hope you all know what a dicotyledonous plant is. You've already done in O-levels, a plant whose seed has two cotyledons. Examine the specimen, which means examine the slide using the low power of your microscope. You'll be using the low power lens, which is either the lens which has the magnification of 10 or 4. You have 3 lenses. The lens which has the magnification of 4, the one which has the magnification of 10, and the other 40. So you can examine it on 4 or 10. And then after doing that, you are supposed to draw a large labeled plan diagram to show the distribution of tissues. Okay, in the last class, I had given you an idea about how different plant tissues look like and how to identify them, right? Now, when we are doing slide questions, which is the second question in your practical exam, you are either asked to draw a plan diagram or a high power diagram, right? Let me just give you a brief introduction of what is the difference or what do you mean by a plan diagram. Okay, so let's start with knowing what a plan diagram is, right? Now, in your question today, you've been asked to make a plan diagram and as well as a high power diagram, right? So a plan diagram is the one which is always made in such a manner that the individual cells are never drawn. That's the hallmark of a plan diagram. Number one, the first point you need to remember is that whenever you're drawing a plan diagram, this means that you're observing the slide under a low power. Always remember this. You're observing the slide under a low power. And such a diagram is known as either a plan diagram or you can also call it a low power diagram. The basic guidelines for drawing any diagram is that you always use a sharpened pencil. The more sharpened your pencil is, the greater are the chances that you will make less mistakes as far as drawing the diagram is concerned. You are not allowed to shade cells. No shading is allowed. Your pencil, as it's sharpened, so then this means that you'll be drawing clear, continuous lines. Your lines should not be broken. And you have to use accurate proportions and not and your own observational skills in drawing a plan diagram. Your plan diagram should not be a textbook version, okay? Your plan diagram should not be a textbook version of the diagram that you're drawing. It should be the diagram that you are observing on the slide under the microscope, right? And what do we mean by make accurate proportions? Making accurate proportions, I'll just give you an example. For example, let me show you this slide, right? Okay, this is the slide of a dicot root. Let's say you are given this slide and you are asked to draw a low power plan diagram. Right? I start drawing with a sharpened pencil like this. Right? Now I can see in this diagram that the cortex of the root is occupying roughly this much space and the central part is occupying this much space. So whenever I'm drawing the distribution of tissue in a plan diagram, my diagram, the proportion of my diagram should be similar in proportion to the slide that I have been given. For example, if I am making the diagram like this, roughly this means that my diagram is very similar to the slide that I have been given because I have drawn it according to the proportion that I see. If instead of drawing these proportions, if I draw like this, it will be a wrong diagram because it's out of proportion with the slide that I have been given. So this is the meaning of drawing a plan diagram according to the accurate proportion, right? Let's go to the same slide where we were, which gives us instructions about how to draw plan diagrams. So we'll be using accurate proportions as per our observations and not a textbook version. Our diagram should not be a textbook version, because as you know, as we keep on doing some more chapters in AS, you will see that there are many diagrams drawn in the chapter transport in plants. So if let's say you've been given a slide, which is a slide of a dicot stem, you are not supposed to draw the diagram of the dicot stem as it appears in your textbook Mary Jones or textbook Soper, but you will draw it as per you observe, because every dicot stem is different, right? It has a different pattern of distribution of tissues, different proportions. Achha, these are some of the rules. Let's do some more rules. Another hallmark of a low power plant diagram is that we never draw individual cells, right? Individual cells are never drawn. A plant diagram only shows you the distribution of tissues. For example, if I were to draw a plant diagram of the slide that I just showed you, in that case, it should have only been drawn like this. I should only show the distribution of tissues. For example, this much space was occupied by the cortex. This much space was occupied by the central part. And yes, in the central part, I could see that the xylem and the phloem bundles were arranged alternately like this. This is how they are arranged. All this is, of course, I'm just, shading in order to show you, but you're actually not supposed to shade, just to show you how the tissues are distributed. This completes your plan diagram for a dicot root. Now, this plan diagram is only showing you the distribution of the various layers, but it is not showing you individual cells. I haven't drawn any parenchyma cell. In fact, I should not draw any individual cell in a plan diagram. This is a correct plan diagram, right? Another thing, we are supposed to draw all tissues completely enclosed by lines, showing that, separate layers. Draw a correct interpretation of the distribution of tissues, right? And if the examiner asks you, asks you to only draw a certain portion, sometimes he might ask you only to draw a quarter of the plan diagram. So in that case, you'll only draw a quarter, right? Sometimes he asks you to draw half a circle. So in that case, your plan diagram, the shape of your plan diagram will be half a circle and so on. Depends upon whatever instructions are given to you, right? You will use a clear sharpened pencil. Your diagram should not be like this. See the feathery lines. Your diagram should not be like this. No overlapping, no broken lines, no feathery lines. Continuous, clear, sharp lines showing distribution of tissues and with an accurate proportion, right? Yes, the other kind of diagram that you might be asked will be a high power diagram. A high power diagram is very different from a plan diagram in that when you're told to draw a high power diagram, the first step will be that you will use the lens with 40 times magnification. This will enable you to zoom into the individual cells and look at the cells very carefully. Depending upon whatever instructions are given to you in the question, you will draw a few representative cells, whichever you're asked to draw. If you are given a slide of the plant cell, you will always draw a double line, and that double line, as I have already told you, it represents the cell wall. A cell wall is always drawn with double lines. Why? Because it's thick, right? If in a plan, if you're asked to draw a high power drawing, and let's say if you can also see a nucleus in your drawing, right, you will draw the nucleus just like a small circle, but you will not draw it as a solid blob. For example, let's say you've been given a slide of onion and you're supposed to look at one of the onion cells and draw a high power diagram. So this is how you will draw. As you can see in the picture I'm drawing on the screen, okay? I'm trying my best not to break lines. As you can see, I'm rubbing and redrawing it once again. Okay. Okay. If I'm asked to draw a high power drawing of an onion cell, there is a possibility that if the slide is really clear, I might be able to see a vacuole. So yes, I can draw a vacuole. And if I can see a nucleus, I can draw that as well. But I will never represent the nucleus as a solid blob like this. Okay, this is a wrong way to draw. The correct way to draw a nucleus is only draw a small circle like this and then label it as nucleus. This is the correct way and this is the wrong way, right? Whenever you're asked to draw a high power diagram, usually they do ask you to label also. Do read the question properly and see if you've been asked to label. Never label a diagram like this. For example, if this is your cell, never label it like this. You can choose one side, most probably you can choose your right side for labeling. So your labeling should be very neat and onto one side of your page only. Another thing is that, let me show you this past paper question and then you will have an understanding. For example, this is your page and you're supposed to draw a diagram over here. Let me go to this part. See, part 3 of this question tells you to make a high power drawing to show a group of 4 cells from the region nearest the center of the specimen. Just to explain you, I'm not drawing 4 cells. Let's assume that one of the cells looks like this. Oh, sorry, I can't draw over here. What I want to show you is that on the screen, you can see a column on the right-hand side, which shows you for examiner's use. So your drawing should not cross this line, which you can see on the right-hand side, because if it crosses this line, that portion of your paper will not be scanned. So that's why avoid drawing in the column which is given on the right-hand side. Draw in the center and look at the question read the question very carefully. If they ask you to make a large labeled plan diagram, then this means that your diagram should be large. And roughly I can say that a large diagram should not be less than 70 millimeters in diameter across. Okay, it should be either 70 millimeter in diameter across, or it can be larger than that, but not less than 70 millimeters. Okay, so we have this question. And this question tells you that you've been given a transverse section, a stained transverse section through part of a dicotyledonous plant. And after observing it under the low power, as the instructions say, you are supposed to draw a large labeled plan diagram to show the distribution of tissues. Now, the slide that you have received today is this slide. It is, yes, it is this slide, right? I'll just zoom into it. Okay. You can see that although, many plants are dicot, but it's not necessary that every dicot stem looks the same as another dicot stem. There are different species of plants and their transverse sections, they also look different. Acha. Now, if I look at this transverse section, which is stained, I'm zooming into it so that all of you can see very clearly. At a glance, I can say that this is a stem. The reason why it's a stem is that if it had been a root, the diagram should have been like this. A root always has xylem and phloem arranged in the center. As you can see, I'm coloring the xylem red so that, just to make it obvious. So a root always has alternate patches of xylem and phloem. The one in blue is phloem and the one in red is xylem. Whereas if it's a stem, it will look like this. As you can see over here, a stem is always hollow in the middle. That's one of the hallmarks of a stem. It's always hollow in the middle, right? And in the stem, the vascular bundles or the xylem and the phloem are arranged in alternate bundles. For example, you see a patch of vascular bundle over here, another patch over here, another patch over here, and then here. And then let me show you some other patches, smaller patches over here, one large patch over here, and so on. If I were to draw the transverse section of a stem in a diagrammatic form, it looks like this. You must have seen this diagram in O levels as well, right? This outer part is your phloem, okay? And the central part is your xylem, okay? And let me use another color, let's say green. This part is your cambium. The cambium may or may not be visible. Actually, cambium is a layer of cells, a layer of living cells, which as they grow, they differentiate into phloem and xylem. Okay, so here we can see a stained slide of a transverse section of a dicot stem. Sometimes we also give the name square stems to this slide. It looks like a square, okay? So if we go back to the past paper question, it says draw a large labeled plan diagram to show the distribution of tissues, right? You're not supposed to draw the individual cells. You're supposed to show the distribution of tissues. Okay, let's see how you are supposed to draw a plan diagram. On the left-hand side, and I'm zooming into this diagram, you can see it's a screenshot or it's a picture from my own slide file. I have tried to draw a plan diagram of the slide that has been given to us, okay? This should be roughly the shape of your square stem. The shape might vary. That's not an issue. Okay, it depends upon, which slide you've got. Mostly all slides are similar, but sometimes the shape might vary. So that's not an issue. But roughly your slide should look like this. You can see that my slide is not crossing. It's not crossing this line. It's within the confines of the space that I have been provided. And if you compare my diagram, let me just rub it. And let me rub this as well so that you can see it more clearly. If you compare my plan diagram with the photo micrograph, with the diagram of the slide that you've been given, you can see that it's more or less the same. In the center, you can see I've drawn a black line. I have deliberately drawn this black line to show you that initially I had drawn this plan diagram with this line. This was the wrong way to draw because it's not according to the proportions that I can see in my slide over here. The correct line would be the one which I have drawn outside, this one. This is the correct because this is as per the proportions that are visible over here, right? So then, as you can see, I haven't drawn the individual cells. I am only showing the various layers. So let me just zoom into the diagram and show you how you're actually supposed to draw it. Okay. Okay, so the outermost layer was the let me use a thinner pen. It's the epidermis, as thin as possible, right? Achha. After the epidermis, then there is this patch. This patch you can see. So this, I have represented this patch. This one, that's a separate layer. These cells are not similar to the epidermis. So then this means that these are some separate layers. So I have to show this layer on the plan diagram over here, which I have shown, right? Then I also have to show this layer, which is the vascular bundle. And then I have drawn the vascular bundle, the shape of the vascular bundle, just to show you the distribution of tissues and not the individual cells. Okay. So the outermost layer, then a separate patch over here, and then this patch for the vascular bundle. You can see this, and then this, and then this. This is your phloem. This is your cambium, and this is the xylem, right? Some of the vascular bundles are bigger and some of them are smaller. So those that are bigger, of course, they will be drawn bigger as per the proportions that you see. And those which are smaller will be drawn smaller. Now the question arises, if you get such a question, how many vascular bundles you will draw? If you've been asked to draw a complete plan diagram of the entire diagram, then you should at least draw 3 to 4 or 5 vascular bundles. I think that's more than enough. If you've been asked to draw a section, then you will draw as many vascular bundles as are appearing in the section. Now, our second task is, just for your knowledge sake, just to revise the tissues that we did last time, was let's identify the different kinds of plant tissues. If I zoom into it, it's not that clear, but I'll try to, make it as clear as possible. This dark patch that you see over here, this is actually collenchyma. It's not so obvious, but it is collenchyma, right? Acha. These cells, these ones, they are parenchyma, okay? And yes, this is the vascular bundle. Again, we can see some parenchymatous cells here and there. Okay, so we have parenchyma, we have collenchyma, and there's one more thing. It's not so obvious over here, but maybe, if you look at the slide, maybe in your slide, it's more obvious, because as I'm zooming out, the resolution of the cells is better. So it can be seen that every vascular bundle has a cap or has a layer of sclerenchyma at the top, if you look at it very carefully. For example, I'll just draw and let you know that, let's say, if I'm drawing this vascular bundle, if I'm drawing this to show you various layers. So this is the outermost cap, which is the sclerenchyma. Then you have your phloem. Then you have a little patch of cambium. And then you have the xylem. So this is how you will draw the distribution of tissues, okay? And you're asked to, let's go back to the question, what are you asked? You're asked to draw a large labeled plan diagram to show the distribution of tissues. So if I'm labeling this plan diagram, I will label it like this. For example, let me use my pen. This will be epidermis, okay? Let me label in the correct direction. So this will be the sclerenchyma. Phloem. If you can see cambium, it's well and good. If you can't, then you simply label xylem, right? And that's about it. If you want to label parenchyma, you can label parenchyma as well. Let's compare our answers with the mark scheme. This is your mark scheme. Okay, these are your answers. 2A part 1. The mark scheme points are 5. It says corner vascular bundle should be bigger than the other vascular bundles. We did that. 3 layers of cells represented plus cambium. No individual cells should be drawn. 4-sided shape, which means it should be a square shape. Xylem in the corner labeled. Phloem labeled correctly. Parenchyma labeled correctly. Sclerenchyma on the outer edge of vascular bundle labeled. Collenchyma in corners labeled. Now see, as you can see, this question is of 5 marks, I think. So any 5 of these points, if you write down any 5 of these, you can get the marks. It's not necessary that you mention the name of each and every kind of cell that you see. Whatever points that you can see as per the marking scheme will be taken as a valid point, and you will be given credit for that, right? Then the other question that they are asking you is, the next question is that state from which part of the plant the section was taken. So yes, this is the stem, and the explanation that we will give should be that the vascular bundles, the vascular tissue is arranged in bundles, okay? And we can see xylem, cambium also. Trichomes are also present. Basically, even if you write down 2 reasons, that the vascular tissue is arranged in alternating bundles, right? And there is the hollow tissue from inside. The tissue is hollow from inside. That's also taken as a correct point. Basically, 1 mark is for identification and 1 mark is for reason. If you want to see what trichomes are, I can give you an idea. Trichomes are not so clearly visible over here. Trichomes are actually hair-like structures, okay, which arise from the epidermis, but they are not visible in this slide. Maybe they might be visible in the slide that you've been given. Let's move on to the next part of the question, and that is for the high power diagram. Let's go to that part. Okay, the last part is make a high power drawing to show a group of 4 cells from the region nearest the center of the specimen. Make a high power drawing to show a group of 4 cells from the region nearest the center of the specimen. Let's see what is the center of the specimen. Nearest the center. Now, nearest the center of the specimen are these cells. These ones, these are, and these are all parenchyma. So this means that the examiner wants you to draw 4 adjacent, clear, high-power drawings of parenchyma. And this is how you will draw. Let me draw and show you. Neat, labeled. Usually, as I told you, parenchyma cells don't have a specific shape. Sometimes they are polygonal, sometimes they are hexagonal, okay? So whatever shape you see, let me draw it in a better way, just a minute. Four adjacent, right? And look now, very carefully look how I will draw this cell wall. Again, every plant cell has a cell wall. So every plant cell will be represented by a double line. The thickness should be the same throughout. You can see that's why I'm rubbing, and so on. Special emphasis on drawing this part correctly. In the same manner as I have drawn the junction of 2 cells like this, the same thing should be repeated here and here accurately, and this completes your high power drawing. You're asked to label it also. Let's go back to the question and see. high power drawing to show a group of 4 cells from the region nearest the center of the specimen. Labels are not required. Okay, so if labels are not required, then this means let's go and see the mark scheme. The mark scheme says, quality of drawing, that is clear single lines, 4 cells similar in shape and size, thin cell walls, 1 line or 2 close together, 5 to 8 sided polygonal cells, air spaces not tight fit, which means you can show a little bit air spaces because we all know that there are air spaces between parenchymatous cells. So that's about it. You can listen to this video of mine, video plus audio, and you can do the questions accordingly, right?